way that I would look at it is, what is driving precision medicine today? In oncology, which is my specialty, I think clearly genomics is making tremendous strides. There are exquisite examples today where we're able to identify and be in a very great position on how to treat a patient simply relying on genomics. However, you got to flip your coin. There's still many instances in oncology where we do not fully understand the biology and how these individuals are responding to their treatments. That's where I see now proteomics filling a very critical piece of that information. So what we're trying to do at the NCI through our initiatives is to begin to converge these disciplines. And quite frankly, partly, I think the reason has been is the evolution of technology. Genomics technology has matured and is still maturing at a faster rate. But as proteomics technologies are also maturing, and also we could go after smaller samples, it's just a matter of time that these disciplines do begin to converge. So that's why I see these two areas being very complementary to one another, specifically in the area of precision-based medicine for all. Proteomics, the way we know it today, would not exist without mass spectrometry. Pure and simple. Genomics gave us a blueprint. What people did within the specialty is, there was an acknowledgement, here is a mass spectrometer. Mass spectrometry has been used in clinics for over 30 years for different purposes. It's mainly used for small molecule screening. But then there was a brilliant idea that came into play. Can you take a big protein, break it apart into small pieces, into these peptides, and informatically, just like an eloquent puzzle, stitch it back up into a protein? Without mass spectrometry, proteomics as we know it today simply would not exist.
As part of the Human Protein Atlas project, we've been making a cell atlas, a subcellular map of the human proteome, using an antibody-based approach. The most interesting findings we made here is that as much as half of all proteins localize to several places in the cell. And we also see a lot of single-cell variation. As a young scientist, I would focus on the integration of different omics technologies and even assays that cross dimensions. So, for example, to integrate sequencing data with proteomics data, with imaging data at both the cellular and multicellular level. I think that's a dimensional challenge that we'll have, and I think that's a very interesting one to tackle.
That proteomics is going to make an impact in the clinic multiple ways. The first way, especially in acute diseases where you're not really looking at gene changes or the genetics except as a risk predictor, I think it's really going to be the proteins and metabolites, the very fast-moving changes that are going to be the ones that we can go after. And I think we already know examples in where it's been protein-based have been the biomarkers in these acute diseases. The challenge comes often with chronic diseases where you're going to need to look across multiple omics to really refine where that individual is and how they're going to respond to therapy. When we do that, and with proteomics that we're doing, we're going to need to look at isoforms and post-translation modifications and other dynamics that will allow you to have really the breadth of response that you need to be able to have to determine that individual's response. We're using targeted mass spectrometry or targeted multi-ELISA platforms to really push into the very large clinical utility space which is needed for adoption.
When we learn about proteins, I think we should try to study the proteins as they are. And so that's my answer to saying, okay, we should do native or top-down. To do top-down proteomics or middle-down proteomics or glycoproteomics, it is very clear that the conventional workflows are not optimal yet, especially in the field of sequencing for glycopeptides and for intact proteins. We don't know yet how to do that best, but we know that CID, collision-induced dissociation, is not sufficient by itself. And that's why we and others invest a lot in electron-based fragmentation techniques, but also combinations of ETD and HCD. And we have shown that this so-called ETHCD is really beneficial, not only for glycopeptides, also for cross-linked peptides, for MHC peptides, but also for intact protein.
Methods are already used in clinical applications, but to measure small molecules. Now, when it comes to proteins, there are additional challenges. And the first challenge is the dynamic range of protein expression, which covers several orders of magnitude in biological fluids and also in tissues. And this somehow precludes identification of low abundance proteins, which very often are very interesting targets for biomarker as biomarkers, as potential biomarkers. The second problem is that to implement targeted proteomics in a clinical setting, you need a sophisticated instrumentation, mass spectrometers, and you also need a highly trained personnel, so people that can run these instruments and that can also deal with instability issues, for example, of the nanochromatographic system coupled to the mass spectrometer. And the third problem is that SRM assays don't have yet the same throughput of ELISA assays. So I would say these are the limitations. But on a positive note, I think as a community, we have already succeeded in demonstrating that targeted proteomics has very important features that are crucial for clinical applications. And these are low coefficient of variation, and also the interlaboratory variability is very low. So this is very important.
All right, very good morning, everyone. I hope you're all excited to learn more about proteomics. You just heard about some of the key experts of the proteomics field, some of our colleagues from Human Proteome Organization. And the idea is, very simple, that we have to study protein. I request everyone to please mute yourself. But the challenge is that, it is not straightforward to study proteins. They being so dynamic and having a lot of modifications. Technologies are still evolving, and we are still trying to understand how best to overcome these several challenges of acquiring the best data set and also analyzing them, also overcoming the reproducibility issue. And newer technologies are keep evolving, which is giving us some new ways of looking at data. At the same time, it also poses challenge about how to take that data and process that data. So therefore, today we are going to discuss more about, I'll show you some workflow of mass spectrometry based instrumentation, how some of the parameter and setting being used before you set one sample for acquiring data of your mass spec. Data search is crucial. We talked last class as well, where a simple data set was given to you, which was essentially, let's say, from SDS page gel or a 2D gel. You just cut a spot or a band, and you want to identify that protein. So that was being done using MaxQuant. But now let's say you have a complex data set, which is coming from a shotgun proteome analysis, and you want to analyze those, and especially you still want to know right now that what are those hundreds of protein present in my complex sample, not just in one band, but in my entire lysate or tissue or sample type. So that's where this MaxQuant analysis will be done today as the second exercise. You will be careful of looking at those parameters and those settings, and you will do some analysis yourself, which will be part of the quiz 2 today. Some of you had reported some issue in the last time of quiz submission. Please start making yourself accustomed for doing that, and don't take the last minute. 20 minutes we are giving it extra time. In fact, 15 minutes what we thought should be given for this quiz. So please try to finish in 15 minutes time so that you have some time to close your stuff and you are not requesting us to redo those things. This will be essentially compensatory quiz. Those who have already done it last time need not to repeat. But those who felt that they have not done very right or they can do something better, they can also do this quiz again. This is not the same quiz, it will be different quiz. Just to make sure that you are clear, it is going to be a different set of file which you have to analyze, but that will be done at the end. So some of you who might have joined a little late in the course and may have lost the track of where we are and what we are talking right now. So we started with proteomics introduction, basics of mass spectrometers. And now we are talking about how these type of mass spectrometry based concepts and data analysis being done. Last lecture and today's lecture will be focusing on this. And then we'll move on to take proteomics to the next level, which is not discovery only, but also quantification. You want to compare your control versus disease sample, or you want to look for the control versus treatment. and you want to find out what are all quantitative changes that are happening. So therefore, you want to do the secondary data analysis of mass spectrometry based work. And that's where MetaboAnalyst, Perseus, these kind of software will be shown and discussed. We'll then move on to look at the biological significance of these data set, come up with more pathway analysis, network analysis, using reactome, string, human protein atlas. And once we have understood some biology part of it and we want to be sure that we are looking at the right proteins and peptides, we have to validate those using targeted proteomics, and again set of softwares like Skyline, Panorama, and Export, those will be used. So this is the each one of these are going to give you some software flavor and some hands-on exercises to be done during the class time itself, and that will give you a quiz from at the end of each of this exercise. We'll also talk about proteomic sample prep and quantification, which will be last to complete this type of work. Additionally, I'll talk to you about gel-based proteomics, but rather much shorter time is being spent on that. And we will initiate some proteomic data analysis projects with all of you. Those will be continued even after post midsem. I have highlighted few lectures only which will be used for the midsem, which will be theoretical lectures. And all of the other lectures will not be part of those midsem, but rather we will continue with the class activities and quizzes, so that you are learning and you are practicing more data analysis skill. All of that will be utilized in the project, which will come back to you. So please do not learn and forget that. You have to continue practicing those. And then based on your project, what you will do, which will be essentially a unique set of data set will be given to each one of you, and you have to analyze those and come up with your interpretation that you are going to present in these presentation slots, which will be quite heavy from the marks point of view. And also, while one student is making the presentation, rest everybody else is writing the summary of what that presentation is about. That will be the write-up component of it in the class. We will also touch very briefly about interactomics and microarrays, and also several revolutions which are happening in the field of omics. Again, this field is very vast and a lot of contents can be talked. Right now, I'm also running an online course of MOOC, Massive Online Open Course. If some of you have time, I think it may not be a bad idea to enroll that. That course will be starting, I think, in a couple of weeks' time from now. That will be much more elaborated in terms of the basic technology like gel-based proteomics, sample preparation. Those things I have talked much more in detail. But as the technologies have really evolved rapidly, I thought for this class and this student, it will be best to get you the best latest flavor of the technology so that you can utilize that in your own projects. Also, we are offering another MOOC course on proteogenomics. If some of you are also keen on learning about genomics and data integration, that course could also be useful. Both of these are free open access online courses, which you can take as for your knowledge and for your assignments. You need not to write exam if you don't need the credits for that. But yeah, overall, I think this field is quite elaborated, and large innovations are happening. Many things are going on. In fact, I offer a full course on interactomics as well. But at some point, you can talk to us, and if you're interested, you can learn about those, how to perform protein interaction, protein drug interactions, using different type of array platforms, using different type of label-free biosensors. But again, this course can only give you a small flavor of these activities. All right, so in the last few lectures, we started focusing more towards mass spectrometry-based proteomics, which is ideally only one of the technology among many which one could use in proteomics. But we are focusing more on it because the way whole field has started moving towards mass spec-based proteomics, I think all of you will be heavily benefited if you learn this part much more in detail. So a simple workflow starts from protein, digesting to make peptides, separating those in chromatography, ionizing those with different type of ionization source like electrospray ionization or MARDI, separating those based on mass and charge ratio using different type of mass analyzers, generate MS data, PMF only, or MSMS data for peptide sequence analysis. For doing so, any mass spec configurations require three major components of ionization source, mass analyzer detector, and three accessories like sample inlet, various processing units, and data output. During this process of any mass spectrometer, the key concepts are to look for the tandem mass spec MSMS analysis, where in addition to two mass analyzers, MS1 and MS2, you also have a collision cell. In collision cell, you are isolating the most intense precursor, which you are isolating from the first scan, survey scan. and then you are generating the fragmentation pattern of that, which gives you MSMS data, and that data you are going to utilize to generate these B and Y ions, which are going to create the peptide sequence for you. So based on these, we talked that there could be n number of mass spectrometer. You don't have to feel afraid about now anymore that what mass spec instruments are being used for that data. Any mass spec instrument, ideally you can dissect them out based on their different type of mass analyzers and annihilation source. So most of them will be having ESI as annihilation source. Some of them also have MALDI as annihilation source. When MALDI as annihilation source, only TOF tubes are linked to that because it is only positively charged, which requires more separation window. So therefore, you only interface with time of flight. Whereas with ESI, you need to reduce that window for M by Z. Therefore, you can use different type of mass analyzer, which are also limitation, but you can overcome that because you are generating multiple charge states. And therefore, you can use quadrupole along with time of flight. You can use different type of quadrupoles. You can use quadrupole along with Orbitrap and ion trap. You can use different TOF analyzers. So all of these are different type of mass spectrometers, which are being used in different papers, which you'll be studying. So what we are doing now from the last class and this class, and we'll be continuing doing this in a couple of more lectures, essentially this workflow which we are following up, where part of it we have shown you some demos in the lab, and essentially now you are focusing about the data analysis aspect of it, how to these chromatogram which is observed here, how to make sense of that data and trying to identify the proteins of interest from that data using different type of database search, right? So this part is what we are focusing more, but we also thought to give you a little bit flavor of the other part of it, like the settings of the instruments. Let me warn you that every instrument, as you can understand, that QTOF will be different, Orbitrap will be different, multi-TOF will be different, triple quad will be different. So every instrument is going to be different. Therefore, this may not be very important for you to understand the settings part of the instrument which I'm going to show you, right? But I'm going to just show you one of the instrument type which we use very heavily and which is one of the state of the art and the best instrument in the mass spectrometers right now. So I'll just use this as an illustration to give you an idea that before starting to acquire the data, you have to pay attention to a lot other details as well at the hardware level and their configuration to acquire good data set, right? But I'm just warning you, you don't have to make it generalized. You don't have to worry about understanding or memorizing these words and these terms and these settings to extrapolate that to a QTOF or to multi-TOF, because every instrument is going to have their unique features and unique settings, right? But what we are looking at here, we are looking at the scan rate of Orbitrap, scan rate of ion trap, 18 or 20 Hertz, and what I'm going to use the more factory settings, which is kind of the default. Resolution can be achieved maximum of 500,000, which is very high as compared to the QTOF, which can go maybe 20 to 40,000 resolution. Therefore, you can understand why Orbitraps are more preferred nowadays for doing the deep proteome analysis. For example, from a given cell or given sample type, you have 10,000 protein, 5,000 protein, you want to capture all of those, so you need much bigger resolution, and that's where these instruments can be much better for you for those applications. If the idea was only to identify the protein of interest from the band or a spot, even multi-TOF or any other multi-TOF, QTOF, any other instrument configuration can be equally good for that matter. You're also looking at in the setting the quadrupole isolation window, the ion trap isolation window. What should be mass accuracy with external calibrant 3 ppm in this case, with the internal calibrant 1 ppm. Different type of dissociation options are available. We have been alluding due to that Orbitrap fusion provides you multiple options. Collision-induced dissociation, high collision density cell, electron transfer dissociation, ETSCD together. It can go up to MS10 for the MSMS analysis. Three different mass analyzers being used. That's why this instrument is known as tribrid mass spec, quadrupole, Orbitrap, and ion trap. Two different detectors are there. For Orbitrap, there's a separate detector. For ion trap, there is a separate detector, right? So this is the one of the instrument. For this, I'm going to show you a little bit more in detail. We will give you some demo as well of the settings linked to that. Now, in one of the previous lecture, when the thermo engineer was here, I showed you the calibration which was happening live that time. For before performing any experiment, you want to make sure your instrument is clean and your instrument is calibrated, because that is going to be very important. You don't want to have any noise coming in, which is going to screw up your mass, and that is going to give you the false numbers. So every component of the instrument from the beginning, like all your peptides are going to be injected here, and they are going to make the spray, which is going to get ionized states, which is going to move inside the mass analyzer. All that has to be very clean, right? So you need to make sure that the sweep cone, which is this part, the ITC tube, which is this part, all of them are extremely clean, and they actually start getting some deposit because your peptides, they contain some buffer component, the salt component. Sometimes you may have some other type of artifacts in there. So whatever you do the cleanup, still there will be some residue errors or certain artifacts which will be carried over, and that is being protected from this part. So first, you want to make sure that those things are clean, and you are going to utilize a good calibration mix to make sure your instrument is well calibrated. And there are some standard calibrants comes for both positive ion and negative ion calibration solutions, which are essentially few known compounds like the caffeine, MRFA, Ultramark. These kind of calibrants mix can be obtained from the commercial vendors. In this case, for the Orbitrap instruments, we have this particular calibrant mix. Again, as I said, some of these things are just for illustration. You need not to follow exactly same catalog number and same detail for every other instrument. Every instrument will come with a different set of settings and its own calibrants. So here, before we start our experiment, we want to make sure instrument is well calibrated. And for doing calibration, this separate type of ionization source is being kept, which is the hazy ion source. And while doing so, then you can inject 500 microliter of calibration mix solution. You are essentially starting with the positive cal mix. Maybe a flow rate, higher flow rate can be set up 5 microliter per minute. And then you are going to start with the the basic settings of a wide mass range of 150 to 2000 m by z, AGC target 1 e to the power 6 and 17,000 resolution. Now, while doing so, there are many instrumentation checks which you have to follow, which we are not going to talk in detail right now, but you will have to make sure every cell is coming green while doing the calibration part. The pressure is not exceeding as per the limits given on that. And all those checks are passing. Like if anything gets failed during this process, then you have to be worried about taking it forward. The stability of these ions during the spray should be stable. And you are looking at total ion chromatogram of those. You are making sure that your residual error for the standard deviation is less than 10% or at least less than 15%. Then only you will find that it is a good signal stability is being observed in your total ion chromatogram. And these are different calibrants which are being seeing, you have specific masses which you want to observe. So again, you want to make sure your calibration goes well. It should be fresh solution made. You are going to do one by one the positive and negative calibrants. You are going to maintain the stability of the spray and all the pressure and all the settings to be followed. So once you have done successfully, all the checks will be shown that everything has passed properly and your instrument good to go. And it generates a report, which is important report to be saved for you for your experiment, because that is what going to give you confidence that all the masses, their efficiency is passed. So to 99 or 100% to that tune. So all these are the known masses which instrument is able to detect, and therefore you are happy with this part. So this setting has to be periodically. You have to keep checking this thing every 15 days. We usually do that. Although I'm showing you one old report here, but every 15 days we do that to make sure that instrument calibration is good and we are happy with the mass. Also during the process, because your tubes, some leaching is going to happen from that, and therefore a specific mass of 445.1 could be used in this case as a user-defined log mass, because this, even if you don't have the external calibrant, what we just showed, this can be used as an internal calibrant, because this mass is always going to be observed. You can also click on this to see like this mass is being observed or not. So these are different type of calibration which you want to do. Again, as I said, you want to make sure first your instrument is well calibrated and also very much cleaned. So your the inside whole of the mass analyzers, all the mass spec part is all protected by the sweep cone, which was in the front, right, which I showed you earlier. Now you want to make sure that this cone is actually kept on very high temperature, almost 450 degrees. And so before cleaning, you would like to make sure that it is actually properly cooled down. You would like to clean them up. You would like to sonicate them, wash them properly. There's an ion transfer capillary ITC, which is as a part of after the ion sweep cone. So this sweep cone and ITC, these are the one which protects your main mass spec instrument, all the mass analyzers coming from any aberration, any sort, any contaminant which is coming from outside. So these are the major protection, and they, of course, very often you will see the debris are deposited on these sweep cone and the ITC. So it is good idea to clean them up at least every week and make sure that you are sonicating them well, you are cleaning them well, and you keep some spare ITC tube, which is, you can replace those periodically and clean the other one. So these are some of the things which I'm talking to you, not as a mass spec, what you generate and analyze data, but equally important aspect before what you start to do any mass spec experiment, because a lot of things goes on in the back end to ensure your data quality remains good. Now, once you are good with your cleanup of the instrument and your calibration of the equipment, now you would like to set up the methods. And now you can go to the method development, the tune page of that. Again, just caution that we are talking about a specific type of instrument, which is from a vendor, which is going to have the specific settings. Need not to generalize this for every instrument and every vendor. Every instrument has some its own predefined settings and interesting ways of looking at data. But in all of those, idea will be to look at what is the scan type you're going to use, MS scan or MSMS scan, what type of detectors you are going to use, what type of scan rate you are going to use, what is the mass range, what is the scan range, what is the AGC target, what is the maximum injection time. So these are different options which are given, and you'd like to play with those options to set for your experiment. And this is what we want to know from this course and this class, what we want all of you to think about, not just, like this mass spec instrument is, how to operate that instrument, but think critically what you want to do from that mass spec, what type of experiment you want to do, what type of data you want to generate. And in the process, whether you can also play with the settings, whether you can also play with the chromatogram, whether you can look for the chromatography to improve that better. And then finally, you acquire data which you can analyze in your own way. I think all that will make your proteomics data much robust. Let me warn you again here, like, people many times feel like proteomics is not as straightforward as genomics. And reason being here is that in genomics, you have like PCR, polymerase chain reaction, where you can simply amplify a gene of interest, or next generation sequencer, where you can sequence a gene. Unlike those, here there is a lot of intellectual aspect is involved at each step, from the sample prep to digestion to the instrument settings to looking at the data chromatogram. So proteomics is not as straightforward. You give a sample and everybody, anybody can generate data for you and give you that data. You have to put a lot of thoughts what you want to get from that experiment. And accordingly, as you can understand from all of these discussions, that a lot of things can be changed as per your requirement. And therefore, you can get very different data from the same sample type, depending upon many different type of settings and chromatography, as well as data workflow which you are going to use. So I'm hoping that you are going to be much better prepared, much more informed about choosing those workflows and parameters for your experiments when you come to doing that. Otherwise, proteomics not like just sample and do proteomics. It's not anymore, right? So you have many options here to do MS analysis, MSMS analysis, multiple type of MS scans, scan range to be chosen. So all of these are options which you can start setting that. And here it shows you different instrument for which you want to change the parameters. Let's say we want to, we will show you this thing live shortly for a live run. So let's say you have set up different sample which are going to be analyzed, and one of that sample you want to see what that setting looks like. So you right click on that, go open file, and that will lead you to these pages when you can see the setting of your LC, setting of your MS and MSMS. So it means for every sample which you are running, in future also you can always go back and go and look for the methods file and go look for the settings, and you can then find out what settings were used. And if you're not happy with that data, you can do the changes in the setting. For example, some of these are default setting, like we are talking about chromatography part, how much peptide to be injected, what is the volume of that, what is the flow rate of that, what is the maximum pressure this particular type of column, NMLC column, can withstand. Then based on these, then you play with the gradient and you look for what should be my percentage A, percentage B, and how much time I want for each one of those, let's say from 5% to 90%, how much time I need for each of those gradient type. And then accordingly, you can see your peptides are being eluted here, and you can adjust those. You can, if you want this part to be coming much early and you want to give more time here for the better separation, you can do that. You can play with this number, make it maybe 25%, and then go to 40%, then go to 70%, like that, right? So you can play with these chromatography parameters to make sure that you get the best Gaussian pattern here of your chromatogram. Then you are talking about, now this thing we talked about, let's say nano LC parameter. Now you can talk about the instrument parameters of fusion. So we can look for the MS and MS-MS settings. Now let's say you click on MS of Orbitrap, and these are the settings which are given. So you can choose at what resolution you need. For some basic LFQ type of analysis, you don't need very high resolution. You can go even up to 60,000 here. The scan range is given here. You can look for RF length, AGC target, injection time. And then likewise, you can go for the MS-MS analysis and look for those windows. Few things like collision energy becomes important. If you are going to use the label-free quantification, or you are going to use the label-based quantification, how well you can do fragmentation of those ions becomes important. And when we will talk about label-based quantification, which includes tags like your I-track or TMT, those tags, random mass tags, then for their fragmentation to generate the reporter ions, you need much higher collision energy, 35 or 32. With the stepped works better over there. For the normal label-free quantification, the 27 to 30 works okay. So these are the kind of thing which will come with your experience. The time, how long you want your chromatography and mass spec to acquire the data, you have to set up that. And based on these, then you can actually drag and drop different settings, and you can make your own methods file here, what is required for making the method for that type of experiment. Finally, after finishing your experiment, you can generate this type of summary data set, which will be very important for any manuscript writing, any report writing, because everything, whatever you have done with that sample, is being documented for your ionization source to mass analyze it, what was the user-defined lock was being used, positive ion calibrant, negative ion was not done in this case, your MS Orbitrap setting, all, each parameter, whatever the detector used, what is the mass range used, everything is provided. And all this is very important for you to document that for any report and your manuscripts in future. So, while the run is going on, you are very curious all the time to see, is that my peptides are coming okay, how well I can analyze my data, and those things you start knowing about it to look at the good intensity of the peptides coming out, overall good chromatogram coming out, and that's what you want to analyze in your data set. Once you start getting reproducible data, you can set up your own methods. And there are different type of manufactured preset methods also available to you to look for your different type of peptide identification, peptide quantification, post-translation modification. You can click on those and they will give you some settings which you can start with. But each one of the sample type, each biological sample is very unique. You have to still refine those settings and you have to improvise that for your analysis. That will be very important. So shortly, one of the TAs, Abhilash, will go to the instrument and give you the demo of this setting, which I just showed you here on the slides. But you will see a live run and you will see quickly the settings of that. After that, Ditru Biswas, another TA, is going to take you and walk you through a software MaxQuant for the analysis, which will be more or less what we have been learning in the MaxQuant, but with the different settings here. We are then going to have the quiz 2, which will be today based on the MaxQuant settings. And then we are going to have the first compensatory quiz at the end of the class. Just to summarize before we move on to the demos and the softwares, I think what's really important in proteomics that there is so much gadgets, so much instruments, a lot of things are there. While I don't think it is really important for you to understand all of the hardware part and so much settings part of the instrument, that's a more technical job. But what is really important that what parameters makes the data output different, that's you have to understand those much more in detail. Where you have to set up the both Orbitrap, ion trap, you have to look for what resolution setting, all that changes is very important for you to understand. Also, the mass spec data analysis is very important. You are getting exposure and you will be appreciating that the different software which are giving you same output, but sometimes very unique type of specific information as well. And their ways of doing analysis is very different. Some of them are commercial like Proteome Discoverer, some of these are open access like MaxQuant and MaxQuant. But the basic concepts of doing MS analysis or the PMF analysis or MS-MS analysis are going to remain same. So I hope that you're all excited to do proteomics, to learn more about proteomics, and you are probably start feeling more comfortable as we go along that despite that you're not in the lab here, you are not able to do the experiments live, but still you are able to get the sense of the data and able to appreciate how these type of data being generated, which we are analyzing it further. So if you have any questions quickly, we can take some questions, or we can also write in the chat. I can talk to you on that. But given that we have many activities planned, I'll quickly request Abhilash to share his screen and take on the live demo of the instrument parameters and settings, so that we can see a live run and see how the settings are being used over there. But please feel free to write some questions and ask more comments in the meantime here. Thank you.
Thank you, sir. So I'll take just 5 minutes to show the LC setting first, and then I'll show the MS parameters. So as sir told, once you purify your protein, or once you have a complex set of proteins, first we need to separate the proteins in the LC setup. Then the peptides will be fragmented in the MS, and where we need the HCD and CID and different types of dissociation. So if, as you see, you might have aware of different types of chromatography, right? So for chromatography, we need 2 different phases. One is the mobile phase, other one is the stationary phase. So since we have peptides and we are running in a positive mode, we need a very specific kind of column that we call as C18 column, that I can show. Okay, so this is how the column looks. So we are injecting our protein or our peptides into this column, where this will try to separate the peptides. But before doing that, how we inject those things? So for that, we have the liquid chromatography here. So as you can see the instrument, we just put our sample. Okay, so we'll just put our sample, then auto sampler will pick. We have to mention the vial position, then auto sampler will take the sample, and it will be separated on the basis of a gradient. So for that, we need 2 different things. One is
Vilas, we can't see your screen or anything.
Can you see the instrument live, or you cannot see the instrument?
Yeah, we cannot see
the instrument, right?
No, we cannot see it, right?
We can't see anything.
It is visible for me.
It's visible.
Yeah, it looks very different because we can see that. Maybe Abhilash, please unshare once and share it again.
Okay.
Just once so that everybody can get. But right now he's not sharing the screen, he's showing the instrument live right now. So share and share.
Okay, so just I need to show you the chromatography thing. Okay, so we have 2 mobile phase. There is the solution A and
Abhilash.
solution B. Solution A is a basic water only.
Abhilash, 1 minute.
And the
You have to stop presenting, because then you can come in the full screen.
yeah.
No, don't present, just unshare the presentation part, because you are showing the instrument right now, so we want to see you in the screen. Much better now. Yeah, please continue.
It's better.
Go ahead.
Yeah. So once your sample is injected, it will separate on this silica tubes. So if you can see the tube, these are the green colored tubes. It will separate the sample on the basis of the gradient that we set using water and ACN. Since the peptides are most of these peptides are hydrophobic, will pass a gradient of ACN. So now you are eluting the peptide, and after elution, it will fragmented in this zone. So this is the zone. This is the flow where column is treated at 45 degrees Celsius, and after that only the IPC tube was mentioned is connected. So this is how the liquid chromatography works. So once the peptides are injected to the column, then the rest of the things we can play with the mass spectrometer that I'll show now. So at this time, once the column is once the peptides is already separated to the column, we need to set some parameters so that instrument can analyze the data. Okay, so here we have, since we are talking about the liquid chromatography mass spec, we have two different components. One is the liquid chromatography, other one is the Orbitrap fusion. So we have to take the chromatographic method first, where we need to give the amount of sample that is to be loaded and the flow rate. Since this is a very nano column is connected, the flow rate we cannot give more than the 5 or 10 microliter per minute. Fine. Then we have to set a gradient. So what we are doing is we are eluting our peptides, already bonded peptides, where it is bonded to the C18 column, will make a gradient. And it is started from the 5%. Then we have seen that most of the peptide elutes around 30 to 60. So maximum time, that is 75 minutes of total 120 minutes, we have given just at the 30% of the ACN and FA. So at this point, the peptides will start separating. So if I show you one live run, I think one run just finished, but if we see, see, this is how the chromatogram looks. So till this around 30 minutes, we have used only, I think, 5 to 10% of the solution B, or which is ACN. After that, till the 90 minutes, we have used only 30% to elute all the bonded peptides. So we'll get this kind of chromatography where you have the information at the MS1, which is which peptides are there, and second, here what I am moving is the MSMS, where you will see the fragments of that MS or fragments of that peptides, which we can later identify using the MaxQuant, the MaxQuant or PD. Okay, to set this method, we have to give certain conditions to the MS. That is the scan parameter. We have to mention the scan parameter that since we are injecting the ions, we have to give a threshold intensity. We have to mention a scan range that you have to, the mass spectrometer has to scan between a M by Z range of 375 to 175. Then the AGC target that shows how many ions to be taken for a given injection time of 50 milliseconds. So you have to mention a specific time and a specific amount of ions that is to be detected. So that setting we can put here using this method editor window. And the DDMS later, the MSMS setting. So for that we have to mention the isolation window, which specifies that what should be the width of a specific chromatography. Then there are different things, and the most important one is the collision energy. Since we are fragmenting, what should be the percentage of nitrogen gas or the gas that we are using for colliding the fragment peptide, that should be mentioned here. We have optimized for our setting, it is 30 and it is working fine for us. And then the resolution of the Orbitrap. So here we can see two types of resolution. One is at the OT level, MSOT, where if you see the resolution is kept very high, that is around 60,000, because we'll see similar kind of peptides having similar length will come at this stage. But up in the DDMS setting, in the DDMS setting, you will see the same from the same fragment ion, you will see a very distinct types of fragments that are generated. Hence, we don't need a very high resolution at the MSMS. But for MS, since most of the peptides may have similar sequence or may have one change or two changes that instrument cannot separate, so that we need to scan this at a high resolution range. Okay, so that's all from my side. Now we'll move to the MaxQuant session.
Hope I'm audible. Hello.
Yes, Deep, go ahead.
Okay. So I have just shared one link in the chat box, and the link will give you some files. If you access the link, it will give you some files and folders. And for this particular session, as we are trying to keep the session very easy and accessible to all of you, so I will request not to download the .raw files over here, and just download this one file, which is Homo sapiens .fasta file, and the MQ parameter file. These two files you will download. So I'm telling this thing before so that while I will give you some idea about the software, in between you can download these two files. So these two files, the .fasta file and .xml file, you will download. And please don't download any of the raw files because each of these files are 1 GB. So these has been given to you for your practice after the the class is over, or whenever you will get some time. And the MaxQuant software, I have make it available over here in the .rar format so that you can download it and unzip it. And if someone is facing problem in the unzip, I have also given it into the normal folder, which you can download. Okay, so I will start my presentation, give you some idea about this particular software, and I have shared the link in the chat box, as you can see, so that you can take this thing and you can I have already shared. I don't know why you can't see, but I have already shared the link, right? So now I will come to the demo session, and I will just take a couple of minutes to make you understand that why we are using MaxQuant. So the thing is that from the couple of lectures, you have understood that somehow we are taking the proteins and we are converting the proteins or digesting the proteins to peptides. And then finally, we are injecting these peptides into the mass spec. Just now, Avila showed that how the columns are there and from where the peptides are getting injected. And finally, these peptides are giving some spectrum. So this is the thing that is happening before we are starting the analysis, right? But what the search engines or the algorithm, even the mascot we learned in the last class, what it actually does is that it is taking the spectrum files and it is trying to give you the information of the proteins. That means whatever the peptide information, protein information that you are injecting into a peptide sample you are injecting into the mass spectrometry, it is taking all these things and it is giving that which proteins are present in your samples and on the basis of the identification. So that means there is something going on in the behind or the back end of the search engine that we need to understand. So to make the things easier for you, I will just give you a very small like a demo regarding the MaxQuant. And before that, we will like to see that how the things are happening. So as you can see, these are the protein samples. Any protein samples you can take, which can be tissue sample, which can be serum sample, plasma sample. And then you are digesting these samples, which is giving you peptides. And these peptides are getting injected, which are giving you MSMS spectrum. So till this part, it is all depends on the instrument and the sample preparation. After this, when you get the spectra, which is in the form of .raw files, which I have just showed you that these are the .raw files for each samples. So this particular raw files contain this MSMS spectra. So now you need to understand and decode these MSMS spectra because most of these files are are not readable files, and you cannot read those files in the Excel or something, which you will get an idea. So these things need to be processed and analyzed. So as you can see that this particular files which are present over here. So please don't worry if you are not able to download the MaxQuant parameter file or anything. Just listen to the session and the demo what I am taking, because none of these parameter file anything will be required for the quiz today. These are all for your understanding and practice. So just concentrate what I'm telling. So now for the MS spectra, whatever you are getting, you need a database to understand that which spectra belongs to which peptide. That means there is something, a background database is needed, which will help you to align your spectral information and identify your peptide. So this particular database is all the proteins of a human. For example, if I'm taking a human database, that means it contains all the proteins of the humans and all the peptides of the humans. And then on the basis of these, the alignments happens. That means the spectral information is aligned with the peptide database, whatever we have given, and it tries to identify the peptides. And whatever spectral information is not available for the peptide database, it not found in the database. And after taking each of the peptides, it tries to align with the complete FASTA sequence of the proteins, and it will give the protein identity. That means it is going reverse from peptides to protein. To make the things more simple for you, I will try to give you an understand of this part, where this is the peptide, for example, that we are getting a MSMS spectrum, right? So this particular peptide digestion happens using trypsin, right? So trypsin is an enzyme, and you guys know that the digestion of the peptides happens due to trypsin, and each of these peptides gives an experimental spectrum to us. So now the database which we are downloading from the UniProt or any kind of any kind of repository, it also uses trypsin as a theoretical trypsin, and it's give you a theoretical spectrum based on the theoretical cleavage of these proteins. That means now we are having an experimental spectrum and a theoretical spectrum. So now it will compare between these two. The experimental spectrum will compare with the theoretical spectrum, and it will give you the score. That means which particular peptide is having the maximum comparison. And on the basis of that, it will choose the best match, which is a 3. For example, for this is the 3.5 is the maximum. And like this, you can understand all these peptides will come and best match peptide will be taken. And then it will get aligned to the whole FASTA or the whole sequence on the protein database. And then one proteins get identified. So over here, one thing is pretty clear, that more the peptide identification will happen, more the protein confidence of the protein identification will happen, right? So from this two information, you understood one thing, that this particular analysis requires one database, and without the database, we cannot do the analysis. So for downloading the database, we use UniProt. So UniProt is a repository which contains all the information available of a particular protein in this world. And there are two important sections of the UniProt databases. One is SwissProt, another is Tremble. So SwissProt is something which is a manually curated database, that is a reviewed database. However, Tremble is something which is a computationally curated database. It's not it's an unreviewed database. So generally for mass spectrometry purpose, where we want to get some confident protein identification, we generally go for SwissProt thing. But there are cases where people take both SwissProt and Tremble to get more identification, more hit for the proteomics. So if you want to download one particular database, you go to UniProt, write human, and over here there will be one particular human protein databases will come, or any species, whatever you want. Click over here and download the FASTA canonical format. So in this particular folder, I have already given this particular thing for you, so that you can download this, and if you want to run the analysis, you can run the analysis whenever you have some free time. Right? So once the database is there, you are clear that you can do some of the analysis. So now coming to the MaxQuant. So MaxQuant is a search engine that you have learned. MaxQuant is a search engine which can do the analysis for high resolution MS data and the large number of files it can run, large number of raw files it can take, right? And for downloading these files, you can go to the MaxQuant link that I have given already, and you can download this particular thing by registering your email ID. And if you are facing some issue, I have already given the software in the folder. So once you are downloading this MaxQuant, and it's a very easy thing. Once you are downloading, it will give you a MaxQuant folder. And if you are clicking over here, so it will have an application, the MaxQuant application icon. Click into the application icon of MaxQuant, and you will get the particular window, something like this. So I will directly go to the the software interface. So the software interface looks something like this. But make sure when you are doing this particular analysis, you are having a particular, this kind of folder which will have all your raw files. So for example, I have 6 raw files over here, 3 tumor, 3 control. These raw files are the information of all the spectral information that the peptides I have injected into the mass spectrometry. This is the database file, and this is the MaxQuant parameters file. So I will talk about the MaxQuant parameter file, but make sure to run one MaxQuant analysis. We should have all these files and at least 2 raw files. If not 6 raw files, at least we have 2 raw files to run the analysis, right? So please don't worry, don't worry if you are not able to download it. Just see that how the things are happening. It's very easy, not much difficult. So the first thing is that once you are opening this interface, you can see there are a number of tabs over here and there. Here are available. The first thing is that we will go to the file. We will go to load parameter, and I will go to the particular that folder where all my informations are available. So I will go over here, and as you can see, the MaxQuant parameter file is there. So this MaxQuant parameter file is something which will have all the information and the parameters that will help to perform this particular analysis. So these are some residue files are there. I will remove these residue files for the last analysis, and then I will from here I am taking the load parameter file, and from here after that I will load the raw files. So I will again go to that particular folder, and this particular folder, when I'm clicking, see all these raw files are there. So as I told, there are 3 tumor, 3 controls are there. So over here we will click no fractions, because our samples are not fractionated samples. So you will learn about fractionation. Not fractionation is something that when one particular samples were fractionated into multiple fractions to get more coverage of the protein. But over here we haven't done that, and it's a simple data set. So over here we will click no fraction. If we have fractionated data, then we will assign the fractions over here, which is present over here, as you can see, right? So once this is done, once this part is done, the next part is to check all the parameters whatever we have put. So once you are moving over here to the next one, which is group specific parameter. So the good thing about MaxQuant is that most of the parameters of MaxQuant are default only, and there is not much changes required for this particular software. So the next part is in type, we will keep default. So I will just show you which of the parameters you need to check. For example, when coming to the modifications, as you can see, the oxidation and carbidomethylation. And as in the last class, if you have done the MaxQuant, you will remember that there is an oxidation and carbidomethylation need to be added. And the carbidomethylation is something which is a fixed modification, because we are using IEA, right? And once we are using IEA, so that is the reason why carbidomethyl group is added. However, oxidation of methionine is something which is a variable modification, and acetylation of proteins in the N terminal is also a variable modification. So these particular things need to be added. But if someone wants to add some other modification, for example, wants to see phosphorylation, then over there is a phospho STY. If you click into this particular thing, it will also added, and then phosphorylation modification will also get in check. But we will just keep the thing simple, and we will just see the oxidation and the carbidomethylation, which is required. So instrument and first search, these things we will all keep default. Then we'll go to the digestion. So as I told, that digestion is something which is very important here over here, because on the basis of the digestion, the trypsin, when we are putting the trypsin will, this particular option will help for the cleavage of all these the databases that we are putting for getting the theoretical spectrum. So this is very important, and we should know that which particular enzyme we are using in the sample preparation for digesting the particular peptide proteins. And the max missed cleavage is something that means theoretically when we are using trypsin, we know that all the cleavage site whatever it is there will get cleaved. But in experimentally or practically, this doesn't happen, right? Some of the cleavage site will not get cleaved with the trypsin. So that is why we are giving a threshold of 2 missed cleavage. That means if there in some of the peptides where the cleavage is not happening, but still we will take those peptides into account, and we will not give more than 2, because if you are getting more than 2, there will be a chances of false discovery over here. So now level free quantification and the other settings we will keep same. Now coming to the global parameter, this is again an important thing where you need to put the database. So whatever the database is there, I am removing it, and I am adding the database which I am told that I have downloaded from the SwissProt of the Uniprot. So once I am putting this particular database, there is a minimum peptide length I have kept 7. That means the 7 amino acids should be there for a particular peptide to get identified. If we are decreasing these things, then there will be a chance of false discovery. There will be, because small the number of amino acid in a peptide will be there, the chances of having that particular peptide in the whole database will be high. And if you are increasing it to 8, 9, 10, even more, then you will not able to identify all the peptides, and that will decrease the confidence of your data. So these particular parameters are very important. Then we will go to the protein quantification. Over here also we will keep, as you have seen, we are keeping same oxidation and acetylation. And then most of the information, whatever you are seeing, that we are keeping over here at default. So another important part is there that the decoy mode of the revert over here. So decoy database is something, for example, whatever the database we are giving, the software will generate a reverse decoy database. That means another database where all the FASTA sequence, peptide sequence will get reversed, and the software, while aligning our spectrum, theoretical spectrum, with the actual database, it will also align with the theoretical database. So with the decoy database. If the peptides are getting identified with the decoy database, that means you will understand that there is a false discovery is happening, right? Because decoy database is something which is not an actual database. So our peptide should not get identified with the decoy database, right? So this particular thing helps us to help the software to identify and understand the false discovery rate. So this is a very important part, and decoy database, and there is a paper of the MaxQuant which has talked about decoy database, and there is a revert and randomize is there. So revert, as you can see, it will just reverse the sequences, sorry, and whereas the randomize are there, which are randomize the sequences. So right. So now if we go to the performance, and if I click here, show all activities, and over here the number of processor, obviously you will give on the basis of your laptop, your computing power, and we will start. If we are clicking start over here, so you will see that the particular software has started analyzing. So once you are putting load databases, click start, it will start the analysis. So as you can see, the particular running the particular software is very easy, and I have given a small workflow that you need to load the MaxQuant parameter file, load the .raw file, click no fraction, load the databases at this Whisper database, and click start. That much simple this particular database, the particular analysis is, right? The software is. So once the analysis is over, you will get a kind of folders in that particular directory only where the raw files are there. And over there, there will be a combined folder is there. Under that, there will be a text folder is there, and under that, there will be a protein.group. This particular file is there, right? And we will analyze this particular file only right now. So for today's quiz, and also for the today's demo, because this is something I have given a demo session, and obviously you guys can take these particular files forward and practice this, and I will recommend everyone to practice this so that you will understand that how these raw files can be taken forward, and the protein identification is happening for each of these raw files, right? But now I will start giving that how the output file, after analyzing this particular data, the output file gets generated, and how this output file can be taken forward, and we can do some pre-processing of the files, or which call data cleanup, data munging, data this kind of terms. That means we are trying to cleaning up the data, and we are trying to understand that how the data actually looks like. So I will use the demo MaxQuant output file. However, I will request all of you. I think Shashwati has already shared the file over here, right? So Shashwati, have you shared the file for the exercise 2 today? Yeah. Okay. Yes, so the exercise 2, and I will request all of you to download this particular file, the exercise 2 file, that this particular link, whatever has been shared, because on the basis of this file only, we will have the quiz. And I will request that you should, like follow me and try to understand how to look into the file, so that during the quiz time, it will be easier for you to go and start doing the giving the answer of the question without any hassle, right? So the first thing is that what we need to do is that we need to open our Excel file, and whatever the for you will use this particular file, exercise 2 MaxQuant output file, but for me I will use a demo file. Okay, so once you are putting it into the Excel, this file should look something like this. And I just want all of you to at least start from here, right? Because it's nothing, you need to open a file in the in the Excel sheet. Okay, I hope everyone is here and we are starting this analysis. So as you can see, the first column is having a lot of things over here, cons and all these things. So what this is there. So first thing is that what we will do, I will select the whole column and I will do A to Z. That means sorting from ascending to descending. And you can see the first few things are coming con, like contaminants, and then a reverse is coming. So what contaminants is there? So if I go back over here and I will go to the sequence, you will see there is an option of include contaminants. So these contaminants are the database which will have all different contaminants that should come during the sample preparation. For example, your hair, while you are doing the experiment, there might be a contamination of your hair. So the keratin proteins might come. So it will have all the keratin proteins which should be denoted as contaminate underscore keratin. Then you have used trypsin, right? Trypsin comes from mostly from pig. So this pig trypsin is also a contaminant. So like this, there are contaminants, a lot of contaminants are there, which people has been, people has been reported in multiple papers, and then one particular database has been made. So if I just see the total, how many contaminants are there, I will. So there is a total of around 95 contaminants. So in my total number of protein is somewhere 2,818 protein, 17 protein. There are 95 contaminants is there. So obviously you can understand these are the contaminants which will not take it forward. Then the next thing is coming REV underscore. So this is the one which are getting identified from the reverse decoy database. So these are the false discovery that is happening. That means from a 2000 plus protein, there are only 27 proteins are identified, which is from the reverse database. That means you can understand that the false discovery are very less. It is happening. And obviously, when we are talking about this much big data set, this much number of peptides, this much false discovery will obviously happen. So the first thing that I generally do taking this particular file is that I remove all these reverse and the contaminants. Okay. So that means the whatever total number of reverse and the contaminants are there, it should be removed. But make sure you should note it down that how many contaminants are there, how many reverse are there, because it will be easier for you to understand afterwards that actually how many contaminants proteins came in your data set. So the next thing is that so this two filtering thing is there, which we need to do, right? Now the second thing is there. So as you can see, there are many tabs are there. Headers are there, and these all the headers are important. But now we will try to find another thing which is generally comes over here, which is a score. So in AJ column, I think for all of you, there will be a column which is a score column. And over here also, I will request all of you to do A to Z. Okay. And you can see there are -2 are coming. So these -2 are something which are false discovered proteins. So even after, hello.
Sir, am I audible? Hello.
Yeah, audible.
So I cannot find this demo quant file. Where is it?
I told that for you guys, you are having this file, right? Exercise 2 MaxQuant output file.
No, sir. Where it is? Like I am in that Google Drive, but I cannot find.
Not in the Google Drive. We have shared in the Excel, in the chat box.
Sir, I cannot find it either.
Okay, let
Sir,
me share.
it is opened by Google Doc file, not by Excel.
No. So you need
Yeah,
to
it's
drag
opening
it
in the
Excel.
There was a
Sir, it
Google file.
don't
There
give
was a
any
doc file
option
only.
to open it with Excel.
I'm sharing this file once again with you guys as a file only. I'm sharing it with you in the chat box. Now is it visible? Hello.
So it's still a doc file.
That is the thing I'm
how
telling.
do
It is
we
not a
convert it?
doc file. It's a .txt file. And to open a .txt file, you need to drag this file. So for example, I'm showing you once again. So this is the file, for example, you have. So this, as you can see, this is a text document file. The same file you will show over there. So once you are getting this file, just take this file, drag into an open format where the Excel is already open, and just open this file. So it will open in a Excel file. If you are facing some issue, you come here, click the right option, open with, and come here, choose another over here, and from here find the Excel. But the best way I will say is that you can, I think for you guys it is opening like this, right?
Yes, sir.
Hello. Yeah, so that is the thing I'm telling. Open an Excel spreadsheet, something like this. Take this particular thing and then come here and open this file. It will open. Something like this. It's not something a rocket science.
No, move
now
fast.
Is it fine? Now is it fine?
It should open, I think they can open with Excel. That's easiest. Please go ahead.
Yes. Okay, so I will just repeat this part as soon as possible, as fast as possible. So first we are trying to take all the contaminants and the reverse, as I told, and please note down the number of contaminants and the reverse files, the reverse number of proteins that is identified. Then come to the score, which is AJ column, and do A to Z. And you will see there are multiple negative will be there, and these all are false discovered, because you will see the Q value is 1. So most of the cases Q value should be less than 0.05, right? So this particular thing we should also deal with. So now whatever the number of proteins are there, these are the actual number of proteins which we will be taking forward for the analysis. So if someone asks you what is the number of proteins in the MaxQuant output file after filtering the data, so this is the number of proteins.
So can you repeat that score column?
So if you are coming to the AJ column over here, the score, and you are sorting based on A to Z, like ascending to descending, then you will see that there are few proteins that are coming which are having negative score, -2. And the Q value over there are 1. That means these are the proteins which are false discovered proteins, right? Because their Q value is less than 0.05. Right? So these are those 5% proteins which has been identified even after putting a significance cutoff. So that is why the Q value is 1, right? And these proteins we cannot take it forward. So we will remove this number of proteins. So there is a total number of proteins which has been identified as somewhere around 2685, right? So now on the basis of this particular 2685, we can take the data forward and we can do the analysis. And over here, for example, if someone asks you what is the number of which protein is having the maximum number of peptides, so we will see where is the number of peptide is there, and we will put a data A to Z, sorry, Z to A, because we are checking. And so as you can see, there are proteins which are having, which are having 9 peptides, which is maximum, and the name of the protein is this one, P41091, and the name of the protein IF2G human, right? So this is the way we should start seeing the data. And for example, if there is some question in the quiz where, for example, there is some particular column it has asked for, just put select the whole header and search. For example, if it's asking for sequence coverage, check the sequence coverage over here. It has the sequence coverage, and it might ask that which protein is having the maximum sequence coverage. So you will do a Z2A, and the first protein 95.2 is having the maximum sequence coverage, and the protein is hemoglobin P68871 HBP human. Obviously for exercise 2 it will be some different protein, but for this demo sheet it is a different protein. And for answering any question that which protein is that, always use the column B, because this is the majority protein. That means in protein identification, there must be, for one particular thing, there might be multiple proteins has been identified. But on the basis of the confidence scores and everything, the majority ID of the column B will choose the best one. For example, as you can see here, a number of proteins has been identified, but in majority B, there is only one protein identified. That means this is the protein. So this is how you can do that, and it is just, you can play with the output file, and I am pretty clear you will get all the answers correct. It is just keep your mind clear what you are doing, and you will get all the things correct, right? So I think, sir, they can go ahead with the quiz session now.
Right, yeah. So we can actually start the quiz. In fact, if any parameter, any setting, any issue they face, they can always put in the chat as well.
Yeah, I will help.
We will not give answer, but at least walk them through any settings, issues, right?
Yes.
All right, so, well, good luck. We will do the quiz 2 now on the MaxQuant. After that, those who are done, you can finish it off and you can leave. Those which were not able to do the quiz 1 for some reason should stay, and we are going to share you a link of quiz 1 as well at the end of that. That's all for today. Thank you.
Okay, so if someone is asking for the score part, so if you go to the score over here, you will see that over here in the AJ column, the scores are there. And if you do A to Z, we will not take the negative score forward. That's it. That much you should know right now. Okay, so if the issues are too much, if the issues are too much, then I will. Yeah, I will just share even the Excel file of the data so that it is easier for